welcome to the Brain and Transcranial Photobiomodulation Virtual Summit. I'm your host, Dr. Joe Doduro. My guest today is Professor Dr. Zilong Wang. He comes from us from University of Texas, Arlington, where he's an assistant professor of research in the bioengineering department. His forte is novel neuromodulation by transcranial infrared brain stimulation with different imaging. He had his Bachelor's of Science from Beijing University of Technology in Beijing, China. And in 2017, he received his PhD. Along with that, he was also received the Outstanding Bioengineering Student Award for that whole zone of Texas. He's the lead researcher in Professor Heinli Liu's lab that is investigating cerebral hemodynamics, metabolic, and electrophysiologic responses to transcranial photobiomodulation. And by the way, Zilong Wang's score is 72.5% higher than all ResearchGate members. And his field is specifically transcranial photobiomodulation. So I'm just so happy to have you with us, Zilong, or Victor, as they call you. Thank you for...
Yeah, you can call me Victor. Hi, Joe. How are you?
All is well. Now, tell me something. How does a physics student get started looking at light in the brain? How did that happen?
Oh, I have to thank Dr. Liu, meaning when I was a student before my entrance of university, bachelor or undergrad, so I met her in Beijing, and she guided me from there, say you have to select applied physics, which is right targeting on the techniques that she's doing research over here. She said if you study good, and then if you have a good research experience in your undergrad, then I can consider to accept you as my graduate student. So from there, before even I enter into my undergrad, then I start to follow the plan that she suggested. Well, she's already started mentoring me. Then she guided me actually to choose the applied physics, and then she guided me further when I come here to first start with the spectroscopy study, which is the measurement. Like she's trying to strengthen my ability to do research and how to measure the brain signal. Then she gave me this project suggesting me to say, hey, there's an interesting topic, light can cure brain. I said, I didn't believe it at the first time. Then when I tried to read the papers from Dr. GL and try myself to see, to feel the light, the treatment, and to measure some of my preliminary subjects, I feel, okay, this thing is working. And that's how I started. But all this background that Dr. Liu gave me, she trained me, he gave me the background of biophysics, applied physics, and then the basics of measuring the brain, and understand light. With all these backgrounds, solid backgrounds, then I can smoothly start this project.
And definitely you're one of the high level minds when it comes to this field. And I just want to say that I got this little Star Wars Jedi thing going on here. The Dark Knight, you're all dark there with the cloud. I'm over here in the white. Hopefully we're going to get along as we go through this.
That's cool. People will not get boring. High contrast.
So what do you hope that we're going to learn by the time we get done with this, just your presentation? What is going to be the take-home message? Once we review the 15, 20, this is your life's work, but what are we going to learn?
I think the take-home may be light is working by making you smarter, and I'll explain you why it's become like some potential biomarkers I'll find it for you. Then you can understand why light can help from there. Then for potential future studies, we'll be able to...
We're going to get there. We're going to get there. Don't give it all away at this point.
Sure.
All right, so let's go to your slides.
Well, the title is just a brief review about transcranial photobiomodulation. One second, let me change the laser pointer here so that you can see better. It's a brief review about transcranial photobiomodulation research in Dr. Hanli Liu's lab. Well, one small correction for your introduction. I'm not the leading investigator. Well, Dr. Liu is a PI, is a principal investigator. In the grant, I am the key personnel for her.
Key personnel.
Yeah. All right, so do you want me to start with my first page?
Go.
Yeah, sure. So the presentation is about my graduate work and some of my postdoctoral work, which is something called photobiomodulation. And this is, as everybody knows, is a non-invasive photon tissue interaction method that uses red to infrared light to stimulate cellular functions. And this is the general usage of this light. Basically, clinically, people have found that it can improve the wound healing, reduce pain, and cure sleep apnea, a lot of treatment effects. Well, the particular recent studies from Dr. Gonzalez Lima from UT Austin, he suggested that if you shoot this one on the brain, say transcranially application of this photobiomodulation, later we'll call it tPBM in short, this can improve human neurological and psychological conditions. And believe it or not, he has tried more than 300 human subjects to let them play games or cognitive tasks. And so that they found that it can improve human memory and the response time. So I'm a little cartoon over here. Basically, you just simply shoot this light on the brain, forehead, and then the person receiving this light will become so-called smarter. And the way they performed the experiment was playing majorly two well-recognizable cognition tasks. One is called PVT, psychomotor vigilance task. I believe Dr. Gonzalez Lima has already talked about this in his interview, but I'll review this. Basically, on a monitor, there will be a white cross in the middle. Then the subject will be asked to stare at this white cross. Then when you press, when the cross disappears, immediately the subject needs to respond and press some button. In this case, the space button on the keyboard. Then the computer will count the delay time of the disappearance of this white cross and the time when he pressed the space, so that this will reflect how fast this person is response to the sudden change. Well, the second game is called DMS, Delayed Match to Sample, which is a memory-related or memory retrieval task. Basically, the subject will see a 4x4 combination of matrix with yellow and red color, so that there will be a pattern there. So in this case, there are 2 red over here and 4 red like that. Then this shape or distribution will disappear, and there will be 2 choices for the subject to choose, like which one was exactly what he saw 5 seconds ago. Then by increasing the number or the number of red and yellow distribution, then it will become harder and harder. The subject will be randomly deal with different level of the difficulty. Then he has to choose and recall what the real pattern he saw. And the response time, like if he takes only 3 seconds to think or to recall, or takes, say, a minute to think about it, so the time and accuracy will be taken into account to resolve his memory retrieval. Then with these 2 games played, then the laser will be used. Basically, this is a laser aperture. It will be shoot on the brain of the right frontal forehead of the human subject. And then after the shooting of this laser, the game will be played with another time for the behavioral effect, to quantify the behavioral effect of the same subject. Then they have 2 groups of people. One group is the real treatment, on the left figure. Basically, the light will be shoot over here with a 4 centimeter diameter on the right brain, while the other group will be the sham group, what we call placebo group. Basically, the laser will be on, and they will hear the sound and hear the beeps, everything the same, except there is no actual light coming out of the aperture, so that the subject will assume or think that they are under the treatment, but actually they are not. Then the result is plotted as this. The PVT result is left, DMS is right. So as you can see, the pre-treatment, which is labeled over here. The dashed triangle and the empty circle are treated group, the real treatment group, and the placebo group. Before the treatment, their performance in terms of reaction time are very similar. Then after the treatment, they become separated with a significant difference. That means there will be a significant decrease of the real treatment group in terms of the reaction time. That means the light is improving the reaction time significantly compared to the placebo group. Similar observation for the memory retrieval. Originally, before the treatment, they overlap with each other, but after the treatment, they're well separated with a significant difference. That means the light is also improving the memory of the human. Then this experiment indicates us that the light is really doing something in the brain by making people become smarter. But at that time, people have an assumption, say why this will happen. And the theory or assumption was the cytochrome C oxidase in the electron transportation chain. So which lead to the next page, which is called the mechanism of transcranial photobiomodulation. The basic belief was tPBM can photobiomodulate or photooxidize the cytochrome C oxidase located in the electron transportation chain in mitochondria. Then this is a cartoon that I made to help you, myself, to illustrate this more clear. Basically, this is an inner membrane of the mitochondria. Well, there are a lot of complex in it. The electrons will be transported along the complex and be eliminated at the cytochrome C oxidase, which is complex 5 in this chain. This process will consume some oxygen and boost the proton from inner to outer side of the membrane, while the proton gradient will then boost the ATP production from the ATP synthase. And this is how regular ATP is produced through this electron transportation chain. Then in the case of laser stimulation, the light will be absorbed majorly by cytochrome C oxidase, which is the main absorber or photoabsorber in the near infrared range in the mitochondria. Especially on the heme side, it's a major absorption. Then when the light is absorbing that, it will photooxidize this cytochrome C oxidase so that there will be more concentration of oxidized cytochrome C oxidase rather than the reduced state. The oxidized state is more activated in terms of the redox reactions, like reduced or oxidized reactions in the chain. Then this will boost the transportation rate of the electrons along the complex. Then they will be eliminated over here, consume more oxygen molecules, and boost more protons from inner to outer side of the membrane. And the higher gradient of proton will then induce or boost the more ATP production through the ATP synthase. And this higher consumption of oxygen will lead to the urgent need of regional oxygen over here, which will thus boost the regional blood flow to carry more oxygen regionally. Therefore, according to this theory, we are expecting to see after the light stimulation, there will be more oxidized cytochrome C oxidase and more blood oxygenation. But this was just a hypothesis or the theoretical model about this light at that time. Then our job was to physiologically or objectively measure or verify for this model.
Brilliant. Thank you for that model. Thank you.
Sure. So what we used was our expertise. Basically, we are a brain imaging lab using functional near-infrared spectroscopy. Then we expand our functional near-infrared spectroscopy to something called broadband near-infrared spectroscopy. Basically, I'll show you how it will work over here as this cartoon. Basically, we'll have one light source to give light, or give photons in the tissue, and one detector to detect it at certain distance away. And in this tissue, there are two types of optical particles. One is called scatterers, which is the gray color. One is the blood hemoglobins as absorbers. And when the light is shot in the tissue, the photon will be bouncing around like a billiard ball around the scatterers, which are the fatty tissue. Once it by any chance hit an absorber, it will be eliminated. But there will be a lot of them that are lucky enough did not hit an absorber and being bouncing around towards the detector and being detected by the detector. Then once millions of them are shooted, they will form a banana-shaped region, which is sensitive, which is actually a sensitive banana shape of our measurement. And all the photons within this shape will be shooted from the light source and detected by the detector. The detector is a spectrometer which changes the photons or light signal into a wavelength-based spectrum. X-axis is wavelength in terms of nanometer. Y-axis is the intensity of each wavelength. Wavelength here in terms of colors. You can understand like 600 as or 400 as violet, 600 as green. 600 as red, and 800 nanometer as near-infrared red. And then you will form something like this, a spectrum, and the height of this curve is the intensity at each wavelength. And this is pre-treatment. And then, 1 second, because my animation has some, actually it's automatically playing. So then when we do something to the tissue on the left side, as you can see right now, there are small red dots. After we do something, when there is a functional change, there will be more or less red dots, which means more blood will be coming through, or more blood at cytochrome C oxidase will be existing under this banana shape. And this will increase the absorption possibility along this banana shape so that you will have less signal detected, which is this lowered spectrum.
Got you.
And this, sure, great. And this difference between these two curves, the red area carries all the information of the change of concentration of the red dots, which is consists of oxygenated hemoglobin, deoxygenated hemoglobin, and cytochrome C oxidase. The conventional FNIRS uses two equations to solve two unknowns. The two unknowns is oxygenated hemoglobin and deoxygenated hemoglobin. And the two equation is based on two wavelengths along the x-axis over here. Then this equation is a known model called modified Beer-Lambert law. Well, for the FNIRS that we are using right now in the market or in the field of research, people use these two equations to solve these two unknowns. All these R is the source detector separation. This delta OD is actually the your data measured. DEPF is a constant and the epsilon is a constant. They all can be found online. Then the two unknowns is just within this HBO and HB, and they can all be they can both be solved from solving the equation. But now we have three unknowns, but three equations are not enough to solve these three unknowns. That's why we use a linear curve fitting to fit for these three unknowns. Totally there will be 121 equations, and with the known parameters of epsilon, which is plotted over here. Those are known factors, and the curve fitting is performed with this red curve and red curve as experiment, while the black curve as an experiment, while red curve is curve fitting to fit for these three unknowns, which is the concentration of blood, deoxygenated blood, and cytochrome C oxidase before and after the change, the application of laser.
Got it.
Then we'll use this tool that I introduced on human arm first, because at that time we are not sure whether we can detect some meaningful signal on the brain. So we'll start from muscle arm, which is the easiest way. Then we set out like this. We have the light source over here. After a shutter, the light will be shooted in the arm over from here, diffuse, and be picked up at nearby location. Then we use the same laser that Dr. Gonzalez Lima, excuse me, that he reported with the cognition effects. Then we use the broadband light source spectrometer, and we 3D printed an eye-shape probe holder, of course with laptop and optical shutter. Then we measured the effects of HBO and HB and CCO before and after the laser stimulation on the arm. Then we changed a little bit by updating or upgrading the spectrometer into QE-Pro, which is a cool-down CCD with better sensitivity, and the long-pass filter, which is to remove some heat effect over here from the light source, and then put the same setup or upgraded setup on the brain with 3 centimeter separation, which covers the tissue around 1.5 to 2 centimeter beneath the scalp. Then the light will stimulate over here. Then we measure the changes induced by this light. Then this is how we perform the experiment or data acquisition. The treatment, the light treatment, which is tPBM, is on for 55 seconds and off for 5 seconds in each minute, and it will be repeated for 8 minutes. Then accordingly, the placebo will be on and off too, except there will be no light coming out of the placebo. Then our data acquisition of our broadband near system will be on only for 5 seconds when the laser light is off. Basically, because laser light can have some contamination, because the treatment is light and detection also light. Although they are in different spectral range, it will still potentially having some interaction with each other. That's why we, to be safe, we turn off the treatment laser before we turn on the data acquisition, so that there won't be any noise generated in our data acquisition. Then this gives us an interleaved measurement style. At the end of each minute, the 5 seconds will record 1 data point, so that totally will record 15 data points as 2 baseline, 8 minutes of laser treatment, 5 minutes of post-laser treatment. And these two, all the in our data quantification, the baseline will be used as a reference, so that what you see later will be these 13 points during and after the laser stimulation, compared to a normalized or subtracted baseline. This is what we see on the arm, and this is HBO, oxygenated hemoglobin. The blue curve is the placebo, while the red curve is the tPBM, or laser stimulation. Because we cannot call it tPBM on the arm, tPBM, the T calls transcranial. On the arm, we can just call it PBM maybe. So the star means significant statistically difference between the red and the blue curve, and we see a linearly increase of oxygenated hemoglobin during the laser treatment period, which is the shaded area, and the 5 points after in the white region is the post-treatment. And we can see the significance start from the 3rd minute when we turn on the laser, and the effect still remained after we turn laser off. That means the post-treatment effect can last longer than at least 5 minutes, and it's not having a trend to drop off. There's no difference between the two groups in terms of deoxygenated hemoglobin, and there's a similar linear increase and difference between cytochrome C oxidase in the placebo group and the real laser treatment group. So basically, this suggests us that the light can stimulate the tissue by increasing the blood oxygenation and the oxygen metabolism in the tissue, or oxygen utilization in the tissue. And this is having great agreement with our hypothesis, which is shown like several slides ago in the cartoon. So basically, we are expecting to see more cytochrome C oxidase and more blood oxygenation. And that's what we are detecting over here in these three graphs. This is the blood oxygenation, and this is the cytochrome C oxidase. Furthermore, we are encouraged to do this on the brain to expect similar observational results on the brain, because on the arm, the tissue cells has mitochondria. On the brain, tissue should also have mitochondria. In principle, the neurons should have even more mitochondria. So that's why we are performing this experiment, shooting the laser on the same location where the behavioral effects are observed. And with a broadband light source, we are picking up the spectrograph or spectrums at 3 centimeters away. And then, of course, we have the sham or placebo experiment over here. Then we observe the similar observation in terms of the oxygenated hemoglobin and cytochrome C oxidase, that still the linear increment during the laser, well, it will remain there after the laser treatment. Of course, the placebo doesn't change much. Then it's verified that the blood oxygenation and the oxygen utilization can also be increased in the brain on the same location where the behavioral effects were observed, and the physiology biomarkers can be found as oxygenated hemoglobin and cytochrome C oxidase. Then at that time, it was our only group who investigated this, and since it's a new finding, and we only did 11 or 12 people, I forgot, on this finding. Although they have big separation and a small error bar, it's still not convincing to people that if this experiment can be reproducible. Then our recent study from a different operator, this experiment was done by me. The recent experiment was done by Tyrell, who will present you, or who has already presented to you about his paper. He repeated my experiment using my same setup, different location, different group of subjects, and different operator, and got exactly the same reproducible results from UT Southwestern. So that's why it convinced us that this observation was not by coincidence. It was a reproducible experiment or finding.
Excellent.
Yeah. So then, furthermore, we can further analyze the data in terms of plotting another way. X-axis is our change in terms of cytochrome C oxidase, and y-axis is change in terms of the blood hemodynamic factors such as oxygenated hemoglobin or deoxygenated hemoglobin. And then we observe a linear increment or relationship between the oxygen utilization and the blood oxygen supply in terms of the real treatment. But the placebo in green, they are not that obvious. The placebo is the empty fine. If they're gathered around zero, there's no obvious straight line relationship. And for HB or deoxygenated hemoglobin, since it didn't change in the concentration-wise, it's not obvious too. But the oxygenated hemoglobin and cytochrome C oxidase, there's a strong relationship in terms of linear relationship between them. Then we repeat the same plot on our brain finding. Also, we observed that not only oxygenated hemoglobin, deoxygenated hemoglobin shows a negative relationship, also linear compared to CCO, cytochrome C oxidase, which means the more you supply with HBO in terms of oxygenated hemoglobin, like your blood bring more oxygen molecule to you, the more oxygen will be activated. Well, or the other way, the more oxygen is activated, there will be more supply from the blood. Well, for the deoxygenated hemoglobin, they are having a negative correlation in terms of, because this guy doesn't carry any oxygen. That's why it will be negatively correlated with cytochrome C oxidase. Similar observations are observed on HBD and HBT. These two factors are difference of hemoglobin, basically subtraction of the HbO with Hb. Usually people correlate this one with blood flow. Well, HBT is the total hemoglobin, which is summation of these two guys, which is usually correlated or proportional to blood volume. That means blood flow, blood volume also shows a linear relationship with cytochrome C oxidase. That's what we found on the brain. Furthermore, we compared the brain and forearm and forehead results by their conversion or utilization of the oxygen. We divide the cytochrome C oxidase by HbO. Then this tells us that what is the utilization of oxygen and cytochrome C oxidase. So we can imagine there is one HbO. Then the ratio tells us that if you have one HbO supplied in the blood stream, how many cytochrome C oxidase you need to can be activated. Then we see a higher rate on the forehead, which is the red, while lesser rate on the forearm. That means the conversion rate or oxygen utilization rate is higher on the forehead than forearm, which is showing agreement with our known physiology. Similar for cytochrome C oxidase versus blood volume and blood flow. Always the forehead is higher than the forearm. Then this tells us that physiologically, what happened in the single mitochondrion is that when the light is on, it will photo-oxidize cytochrome C oxidase. Then this will bring up the photo-absorption of cytochrome C oxidase. Then it will become oxidized. Then you will pumping and release the neutron oxide, nitro oxidase. Not nitron oxide. This is called nitrogen oxygen, or I forgot the term.
That's NO. You could say nitric oxide.
Nitric oxide, yeah, that's correct. I suddenly forgot how to say this chemical. Then CCO, oxygen reduction to water. Then finally, you will have ATP synthesized, and you will have more oxygenation from the blood in terms of hemoglobin. And these two are what we observed, or what we are supposed to be observed. This is what will happen in one single mitochondrion. And once the light is shooting on the brain, especially in the neuron, the dendrite side of the neuron has tons of mitochondria. So they will all under tPBM. Then once they are working together or being photooxidized together, there will be a lot of ATP produced, and CCO will be drastically increased because CCO are located in each mitochondrion. Then there will be drastic or higher oxygen consumption. Thus, the nearby vessel will then provide you obvious or significant oxygenation from the blood supply. Then we are modifying or editing or upgrading the conventional neurovascular coupling in the field of FNERs. Basically, what I'm saying or illustrating is the conventional neurovascular coupling. Basically, when you have a neuroactivation, you will have several responses like glucose consumption, oxygen consumption, ATP consumption, neurotransmitter release. Those will be increased. Further, you will trigger this cerebral blood flow increment or blood oxygen increment. On top of that, if we have tPBM on the mitochondrion, then it will particularly boost the central two ways by increasing the oxygen supply and ATP synthesis. Further, you will boost the cerebral blood flow and blood oxygenation. That's why we think it can be, like tPBM will be very helpful on the conventional neurovascular coupling by increasing the neural activation and ATP consumption and blood oxygenation. That's why we hypothesize that the light will help the neurons. Later, we'll do some EEG measurement to verify this hypothesis to say if the light is helping the neuron oscillations. But before that, when we are presenting our previous work, people questions like, it's just heat effect. You are hitting your tissue, and that's why you are getting the results. That's why we did some experiments to see if really the observation we observed from the light is from the heat effect rather than the light tissue interaction. So in this, actually we published it already. So in this experiment, we will solely use heat stimulation on the same location where we stimulate on the brain, and then we'll see the impact or response of heat on the metabolic and hemodynamic factors using the same broadband near-infrared spectroscopy to detect. What we do is first we shoot the laser, then we detect what's the temperature that the surface temperature that the laser will induce on the brain or on the surface of the brain. Then we use this black device, which is a thermal stimulator, to generate the heat or the temperature that is equivalent to a laser induction. Then see what change does this heat bring rather than, so this red beam has a certain heat and light. But here it has only the heat portion of this red dot. It is absent of the light. Then if these two are same, that means whatever we observe from the light was due to the heating effect. If they're different, that means the light is really doing something in the brain, and what we observed was not due to solely heat.
Beautiful.
Of course, we are doing the same detection method. Then this is the temperature we observed, and it's arises from 31 to actually 33 to 30 to 41. But don't say this 41 is damaging the brain. This is the surface temperature. So the surface temperature after our recent computer simulation, this 41 on the surface is actually increasing 0.3 to 0.5 Celsius under like 1 centimeter or 2 centimeters beneath the scalp. It's not heating up the brain to 41, the brain tissue into 41 Celsius. But this is what the laser will bring up to 41 Celsius, because one more question is that the 1064 nanometer laser is, people will say is high in terms of water absorption. But if you look at the absorption spectrum very carefully, of course, after 980 nanometer, it increases. That means with any wavelength that is higher than about 1,000 nanometer will be highly absorbed by water. But 1064, there is a decrease, a valley of water absorption. Although it's high, it's comparatively lower than other wavelengths. But on the other hand, 1064 nanometer has less scattering effect compared to the near like 800, 600 nanometer. So when you compare the comprehensive penetration depths, by comparing the scattering effect and the absorption effect, they are very similar in terms of 1064 and the 800 nanometer. So it's not generating way extra heat than the 800 nanometer. The reason why we have this high is because we use a comparatively higher density, power density. We use 3.5 watts and 250 milliwatts per centimeter square as our power density, which is the same as the Dr. Gonzalez-Lima's behavioral effects. Then we do our experiment. Sorry for this jumping up. Now back to our experiment. Then you will be same detection as interleaved measurement, and the thermal stimulation will be mimicking the temperature induced by the laser. Like the red dot is what we measure on the brain. The purple curve will be the curve that this heat generator generates.
Correct.
Basically, yeah.
I follow you.
Yeah. Then the observation is very promising that the brown curve is the laser-induced result, while the green curve is what thermal induces. They are opposite. And they are very, very significantly different in terms of concentration change. Basically, for blood oxygenation, the heat will decrease it during the heat, while it will quickly recover after the heat stop. But they will be very significant different to the laser, which generates the same amount of heat. Then the cytochrome C oxidase, although it's not obviously going the opposite way, it didn't change that much. The significance remains from the, I think, second minute until the end. They are very, very different. That means heat generates almost the opposite effect with the laser, and they are significantly different, especially by one-way ANOVA. The heat will significantly decrease HBO, while heat does not change cytochrome C oxidase. Well, yeah, so this is how we address the heat problem. That means what we observed before was not due to the heat. It's partially due to the light also. Then let's come back to the neurovascular coupling, as we hypothesize that the light is changing or upgrading or boosting the neurovascular coupling flowchart. That's why we are expecting to see the neuron oscillations will be modified or changed by the laser. And this is how we verify it. We do an EEG scan on the human brain with eyes open. Then the light will be stimulated on the right forehead. And then because EEG will be somehow very sensitive to sensations, so we minimize or decrease the power while it lasts the time, so that we make sure the dosage, total dosage is the same, was the same with previous study, except we decrease the power and enhance the stimulation time. Then we'll see if EEG power was increased. And the time of the table of the experiment is placebo is like a flat line, tPBM is a baseline, then shoot for 11 minutes. It was 8 minutes, but now we enhance the time since we decreased the power. So we will have 11 minutes, then we have 3 minutes post-treatment. The EEG data acquisition will be no longer interleaved. It will be continuously along the 15 minutes or 60 minutes data acquisition. Then we'll calculate the EEG power.
Got it.
First, what we do is that we have 64 EEG data collection. Then we do some pre-processing to remove the bad contact motion and the eye blink and eye swipe and the jaw. All the noise will be removed from the pre-processing through ICA, independent component ICA analysis. Then we section the data in terms of temporally, in terms of baseline and the stimulation or recovery period. Then we calculate their spectrum in terms of spectrum-based power analysis. If you can imagine, x-axis will be power, y-axis is x-axis is the frequency, y-axis is the power at each different frequency. Then we'll calculate the baseline normalized power density spectra, and then we average the time frequency map for tPBM placebo and subtract them. Then we image it. Basically, this is the data that we collected, and then we chop into different sections. There is one baseline section, and other sections are during and after tPBM. I'm sorry for this shaking. Then we calculate the power at each time section, and use all this power at each section to divide by the baseline and then average among people. Among people and then subtract between the laser and placebo. What you see over here is the baseline normalized and the placebo controlled power increment of tPBM. We are shooting light here, and this map is given from one particular electrode. Totally, we have 64 of this, and we are extracting one of them to show you an example about what does this time-dependent spectrum look like. X-axis is in terms of time, y-axis is the frequency. The color is the increment or decrease of power. The more red, the higher percentage of increment of power is. The more blue, the more decreased the power will be. And the light, the stimulation covers, I called it tPBMT at that time, but now you can imagine it's tPBM over here. So this much is during the laser, and this much is the recovery. The red means power are increased a lot at this frequency range, which is about 10 to 20, which is alpha and beta band in terms of EEG. Then if you repeat this one globally at a different location, you will observe a very similar observation of this red stripe on each block, and they are all located within 10 to 20 hertz in terms of alpha and beta band. Then furthermore, if you plot a topography in terms of this whole brain power stimulation on different band, then what we did was after the pre-processing, we'll have artifact-free data. We bandpass filter it into different frequency band. Then we temporal section it, just like what we did. This is a time and data. Then after filtering, we just section it. Then we calculate power at each location. Basically, it's baseline power and stimulation power. Then this guys will be normalized with baseline. Basically, we'll divide by baseline and repeat for placebo and subtract them. What you will see will be the results of power increment, global power increment of the tPBM in each minute as topography. From the first minute to 11 minutes, the light is on, then the light is off in the last 3 minutes. This row and this row is the mean increment among subjects. By the way, we have 20 laser, 20 placebo subjects. Then this is the mean, the colorful map, and the blue and yellow dot is the T-map with Bonferroni correction. Basically, it's a significant, if there's a red yellow dot, that means it's significantly increased at that location in terms of power compared to placebo. Then the third row is something called effect size, which is another statistical term to consider the significance of separation between two groups. In this case, it's laser and placebo. So this is what we observe in alpha band. Later, we'll have also observation on beta band, which is following the same processing step, except we bandpass the original data into alpha and beta bands, and we'll also observe that there is a dose-dependent increment, especially in the last 4 minutes, the power enhanced. Over here is also its dose dependency, but the last 4 minutes, the power enhanced. Then the significance are globally distributed, especially after the 8 minutes of stimulation. So does this effect size. That means the light is also modulating the brain oscillations. On top of our previous finding that the cytochrome C oxidase was increased at the local region. And then we believe, according to brain networks, the power or the neural activation propagate all over the brain through neural activations.
Well, 1 second, go back 1 second for this one, please, before we get to it. So if I'm not mistaken, we're looking at this so that if we go to the 1 minute, right, and we come down to the dark blue with the yellow dots, right?
Yes.
We come down one more into the effect size. What we're looking at in the area that is dark blue in the 12 o'clock to 3 o'clock area, that is the left part of the forehead that we're kind of looking at, right?
Oh, no, that's the right side. My apologies. So yeah, this is right side and this is the left side.
Okay, so what I'm asking is you're putting the laser on the right side of the brain, and you're seeing the changes, a lot of them on the opposite side. So you kind of touched on that when you said it's propagated through the entire brain, but because look at the waves down in here, you know, there's almost a total lack in the lower section at 11 minutes, a lack of activation in the right prefrontal area where you're working. So you kind of saw something there that you know kind of needs you to explain it for me. So tell me about that.
So for EEG, it's really hard for us to localize the location activation in the topography because EEG are brain electrical signals. They are originated from different layers of the brain. Most of them are from deep brain. And then because of the conductance of electrical signal, they will propagate from inner to outer side from the conductancy of the circuit. Then somewhere from the back of the brain will be propagated maybe on the scalp and being detected by your frontal scalp detection. So we cannot say the localization on the scalp is the real source of the activation of the signal. So we need to do something called reconstruction or source localization in order to localize where the stimulation or activation is.
I think that's a great explanation. I know people are going to have that question. That's why I put it up.
Sure. And then this is thank you acknowledgement for my PI, Dr. Hanley Liu, and the co-I collaboration, Dr. Gonzalez Lima from UT Austin, and a lot of our group mates who did a great contribution in those work. Thank you.
Ah, there you go. There you go. So well done. Well done. I think that's a marvelous walkthrough of the time that you've spent researching this great field. That one question I have on the neurovascular coupling and the cerebral hemodynamics, because that's kind of like you gave us a good overview of what the near-infrared spectrography is, Victor. But what you're trying to say is that in the normal model, if I'm not mistaken, your brain is going to have some more thoughts, let's say. You're going to activate your brain, and that will cause blood to come in and more oxygen to come in. And they've never really had an experiment, if I'm not mistaken, that says, now I'm just going to make the brain act like it's thinking more. You're not having any thoughts when you put the light on your brain, right? So you're not thinking anymore. You're not pushing anymore, using any more of your brain voluntarily, but you're involuntarily turning on that oxidative process as you talked about. Am I correct in my assumption of how that is a different neurovascular coupling mechanism that you were trying to explain?
Exactly. So this is you're talking about EEG, right?
I'm talking about that the blood flow section that you saw. So first you saw it in the blood flow.
Yeah.
That you're by turning the light onto the head, you activated more energy and more blood into the brain for it to be utilized. And then that's a different kind of mechanism that you pointed out, right? Mm-hmm. And then you said that it's being utilized by producing more of these brain waves, right?
Yes.
And most of the brain waves that you saw going up, alpha, strong alpha and beta waves, are indicative of someone that has more alpha and beta waves are doing what with their brain?
Right. So basically, this can be a good indication about how to use this light to improve those diseases which are lack of or decreasing the alpha and beta wave globally. So that, for example, if some disease, I'm just making it up, maybe Alzheimer's disease people are having a decreased beta wave, for example, then you can use this way in the resting state to manually crack up the beta wave. But I'm not sure whether it's safe to do it or not right now, because maybe some diseases are having an increased beta wave. Then it will be dangerous for us to,
there you go.
To do this. Yeah.
There you go. But we have the ability to understand that the parameters that you put together can change the brain and have actual measurable changes. And one of the only labs that is doing this type of work. I'm thinking.
Right. Or we can say it's just that the light can modulate the brain oscillation waves for now, but we cannot go say any further, say how and in what case, in what way it modulates the wave.
Oh, I got you. I got you. And I think that there was a great further, understanding that it's not a thermal effect. It's not because we're heating up the tissue or heating up the brain. And I'm really thank you for that clarification, because when I was going through it, I was a little dim for me. So I thank you that you actually redid the whole experiment after people, commented on it, to show that if I put heat, on my head, let's say people are going around and say, well, I'm going to put an infrared heat lamp on my head because I'm going to make it a better brain. That's not really true.
Yeah, that's not what happened. Yeah, right. Yes.
And I think that's a great contribution. So
thank you.
What do you see? I mean, your lab is, it's got 10-year projects going on. You have everything planned out and how you're going to work on this. So what do you think is going to happen in 10 years when we're doing the 2030 brain and transcranial photobiomodulation virtual summit? What do you see your field? What do you think your lab is going to be studying? How do you think it's going to be applied to the client, to the human, on the street? What do you think?
There will be, since this is a pretty new field or interesting field, a lot of unknown or undiscovered questions. Say right now, since what we are doing is just 1064 nanometer, we know the peak absorption of cytochrome C oxidase, according to the chart, is not at 1064 nanometer. It's at, I think, 800 or somewhere there. Then it's really worth for us to repeat our experiment methods on more wavelengths to see which is the most effective wavelengths. At least wavelength range, say 600, 700, 850, 980, those are commonly used, right? We haven't used them and compare horizontally to see which is the most effective in terms of physiology changes. And also we just observed this EEG signal change. Again, it's really a superficial understanding. There should be more deeper theories beneath it. We need to dig deeper in this so-called 10 years. And so this, a lot of such of those questions will be waiting for us to discover. And yeah, so we have a lot of things to do, it seems.
I think that your lab and your work is going to be right there, right on the cutting edge as usual. Thank you so much for your time and expertise and all the great work that you and your great colleagues, and especially Dr. Professor Hanli Liu. Thank you.
Well, thank you for giving us this opportunity and this platform.